Okay, we are recording. It's going to be business as usual again. I'm going to give a series of attendance words or attendance phrases throughout the lecture. So to claim credit for watching the video, you're going to need to write those words down. And also every time I say one of those words, you're also going to need to take a screenshot. And then at the end of the video, you're going to email me the list of attendance words along with all the screenshots that go along with it. And then finally, you'll need to send in that email one question about the material presented in the lecture. So that's it. We are about, let's see, it's 7:42. So it's about 15 minutes before the lecture starts. So if you're watching video, you can skip ahead about 15 minutes, and that should take you to the beginning of the lecture. Morning.
Morning.
Do anything for Halloween?
Probably not. Well, you?
No, just the usual kid stuff. Taking my wife taking kids out trick or treating, and I'll stay back and give the other freeloaders candy. I just remember, I guess when I first started college in Tallahassee, I was Halloween was a fun time to be out and about. Just leave it at that.
Yeah. Got a project I gotta do for my study classes. We're doing presentation on topics that we like or know well of.
Very responsible of you. Good. So what's your topic?
United States Marine Corps.
Nice. Oh man, I would love to get that presentation.
Oh yeah, and I'm probably gonna butcher the fuck out of it. I'm gonna be honest.
Why? Because you're just not good at it, or you're nervous, or you don't know the material? What is it?
I have a speech impediment and I have ADHD. My brain goes. So.
Yeah, a lot of times the PowerPoints help me on that. Because otherwise, my brain, and it still does, even with PowerPoints, I'll be go off on a tangent. I'm like, all right, let me get back. I have that problem too. How's it going, Wilson?
Good.
Do you have any Halloween plans? Yep. Morning.
I've been outside now before I just skate.
Oh, came in?
Yeah, I couldn't find it, man.
I sent it to you in that email. That email where it said you could join us online, and it was blue where it said join online. You just click that. I'm sorry, I should have made that more obvious. You can still leave if you want.
Come on, Rick.
It's very responsibility though to not want to bring it and share it. What's the game like this weekend? Where you playing? Who you playing? When you playing?
One of the 12.
We have to win out first of the year to make playoffs.
That's what, 1, 2, 3. 4 more games? Just 3. Oh, all right. Let's do one more, right?
Yeah, but it's the top 3 teams of the conference. And what really shot us in the foot when we lost the rest of the league. Because they were 0-5 when we lost them.
Oh, that sucks.
But they're not an 0-5 team.
Right.
Like they play a lot better than an 0-5 team.
I know what you mean.
So, and I already knew what was going to happen. I was already calling it. Although we smacked Fairmont, and that was supposed to be a game.
Right.
And then we beat Concord.
Yep.
So we play West Lid. We were all hot. It's the track.
Just like all my teams.
No, really good. This is losing the field.
Yeah, and then we lost our Wheeling. But we definitely shouldn't have lost it.
Who was 7-27?
Like, that should never happen.
Was that here or there? Here. Oh, even worse.
Like 17-20 is.
I'm trying to talk my family into going. I don't want to go by myself. But none of my kids or wife want to do anything on a Saturday. So here I am. I guess we go. All right, we got less than 3 minutes, so I'll go ahead and start the announcements. Most of them were repetitive anyway. Turn this off. So again, you can meet me online to get the correct answers for exam 1 and a 5% boost to your grade for exam 1. Do that for the rest of the semester. Obviously, the sooner the better, because that way you can study exam 1 to help you study for the final. As a matter of fact, speaking of which, and I haven't mentioned this before, once your grade is set, once you've retaken it, and I've gone in there and changed your grade, it's helped your grade, you can still keep going back and retaking it. Obviously, that's not going to help your grade, but it'll help you study for the final. Because it's not like you're just looking at the answers, like you're practicing answering those questions. So anyway, that's exam 1. Exam 2, again, you can still retake it until 11/9, so November 9th. You can keep taking it until then. One of your classmates emailed me last night and said it was not open. So after class today, I'm going to take a look and make sure that thing's up and running for you guys. But yeah, you can retake exam 2. Again, unlimited retakes, open book. Let me know when you're done so I can adjust your grade. Any questions about that? All right, exam 3 will be due again, like I just said, on November 9th. It'll be business as usual, 50 minutes to answer about 50 questions. If you need more time, you should let me know now so we can arrange a time to meet online so I can proctor your exam. So any questions about exams at all? All right, again, my office hours today are 11 to 12. So if you need me, I'm happy to help you. I'll be online and in my office. I can give you feedback. I can give you help, whatever it may be. Please let me help you. Any questions about office hours? All right, this week's lab, like I've mentioned a few times, it's completely virtual. It's also pretty simple. So once again, I'm not requiring anyone to be here for this week's lab, but I will be in here if you need me. Of course, if I'm in here and no one else is here, I'll probably wait like 30 minutes and then I'll leave. If I leave, I just need to go to the office. So even then, if you need me, I'll still be online and I'll still be on campus just in my office. So I'm here if you need help for this week's lab. Any questions about this week's lab? All right, pay attention to this. This part's a little bit different. I don't want to say I'm changing things up. I like going back to the original. Next week's lab is also virtual, but it's a tough one. Students generally have a hard time with it. So what I am going to require you to be here next week. So what we're going to do next week when we meet for lab like we usually do, I'm going to run this virtual simulation. We're going to watch it together. We're going to talk about what the data means, and that's how it's going to work. Instead of you doing it on your own, we're all going to have the same data, but then we'll be together to talk about what the data means. So again, next week's lab, we do have to be here, right? Attendance is mandatory for next week's lab. Also, speaking of which, there's a pre-lab for next week. So, again, we're kind of going back to business as usual. For the past couple of weeks, it hasn't been that way. But there is a pre-lab due this Friday for next week. But it's easy. This pre-lab is just doing the 2 videos. Of course, there are some hypothesis questions. If you want to go ahead and do those, you can. I'll consider giving you extra credit for that. And actually, just to be safe, just in case I haven't lost my mind, make sure I'm accurate, right? So I'm saying there's only video questions, but double check and make sure if it's written and says there are pre-lab questions, then ignore what I say. Don't say, you said there was no pre-lab questions, so I didn't do it. Go by what the form says, but I think off the top of my head, it's only pre-lab questions. Anyway, any questions about next week's lab or this week's the pre-lab that's due this Friday? All right. Again, make sure you check your course emails, or excuse me, your WVSU email and your course announcements. Important information comes there, such as, hey, I can't make it to campus today, so here's the link. Speaking of which, one of your classmates brought this up to me. When I do send an email like that, and I say, here's the link, look up the writing. It'll say something like, meet me online here, and that will be in blue, and that indicates that that's a link. So when you see that in your email, click on that, and it'll bring you to the online platform where we can meet. So any questions about that? Okay. Finally, as usual, I'll remind you, attendance is mandatory and participation is graded. As is now the case, you guys all need to send me the words by 9 a.m. I keep repeating that, by 9 a.m. to get full credit. And you have to have one question about the material covered in this lecture. As we've been doing for the past couple of weeks, I'm hoping you guys can get it now. So that is required to get your participation points. If you're online streaming, it's the same thing for you, right? Make sure you send those words before 9 a.m. If you are watching the video, as I already mentioned when I started recording the video, you're going to need to send the words and the screenshots. So yeah, that's a little bit misleading. If you're online right now, if you're streaming right now, you do not need to send the screenshots. You only need to send the words. But if you're watching the video at a later date, you need to send the words. So any questions about any of that? time is it? It's 8:02, yeah. Past time to get started, so let's do it. So if you've missed a class, speaking of attendance, I'll remind you too, there's a whole video. So if you missed the class, if you need the points, go back and watch the video. Write down the words, take the screenshots, send them to me with a question, you get your points. First word for attendance is oatmeal. Today is National Oatmeal Day. All right. So if there's no questions, let's move forward. All right. So we left off about to do the review. I said, now we won't do that. We'll just do it tomorrow. So this is perfect. So really quickly, we're going to review everything from this middle bullet point. This idea of going from RNA to the DNA to RNA to protein. Really quickly, just to remind you of everything you learned on Monday and previously. Just going to remind you, then we'll move forward. So here we go. Again, the flow of information goes from DNA to RNA to protein. Make sure you know that before you know everything else. That's the biggest picture. If you don't know that, then all the other stuff doesn't matter. Like, who cares what it's called when you go from RNA to protein if you don't know that we go from DNA to RNA to protein, right? Make sure you know that. Then once you know that, then start memorizing the names. So going from DNA to RNA is called transcription. Going from RNA to protein is called translation, right? Once you have that, realize that transcription happens in the nucleus, at least with eukaryotes, hence the little star, right? For eukaryotes, transcription happens in the nucleus. Translation happens outside of the nucleus, right? These are things you need to know. I don't know why I included this part now that I think about it. So this is true. For translation, one ribosome can make an averaged polypeptide in less than a minute. I never mentioned that before, so it's not technically a review. And also, let me write this, put a little X through here. Just so to visually remind you, I'm not going to ask you that. You do not need to know that for the exam. That's more like trivial information. Anyway, after that, after translation, the protein, again, it's still a polypeptide after translation. All it is a chain of amino acids. It's not technically a protein until it folds and shapes, or folds and gets into its original or final shape. At that point, we consider it a protein. I'll also mention this, and I don't remember if your textbook mentions this or not, but we didn't really even talk about that bullet point, right? We never even got into how that happens. So if you want for extra credit, you can answer that, look into that. What's the cellular machinery responsible for the folding of the protein, right? You know how transcription works, you know how translation works, but we didn't really talk about how that works. Any questions about that slide? Before I show you a picture of everything that's on there. All right, like we said, transcription's happening inside the nucleus, right? You've got this strand of DNA, RNA polymerase attaches to it, it opens it up so it can read the code. It makes this strand of mRNA, right? Including all the junk DNA, the introns. Once that's done, the RNA polymerase releases itself. The mRNA releases from everything. Then we get rid of those introns. We add a cap, we add a tail. Then it's ready to leave the nucleus. So it leaves the nucleus where, meanwhile, while all this is happening, we have these tRNAs getting attached to their specific amino acid. And I also mentioned this too. This is the first time I mentioned that. So if you're wondering, like, wait a minute, I don't remember him talking about that. I haven't talked about that. But because I haven't talked about that, also, you don't need to know about it. Again, though, a good extra credit opportunity. Because look, I just said, hey, there's an enzyme that attaches the amino acid to the tRNA. So my question is, what enzyme? Is it just like one enzyme for all of that? Or is it one enzyme depending on specific tRNA? Because remember, every specific tRNA has its own specific amino acid. That part you do need to know. Anyway, so we have that tRNA ready to go now. We have this mRNA ready to leave the nucleus. So we just need to bring it all together with the ribosome. That's exactly what happens, even though you haven't seen it. First, you have the small subunit. The mRNA attaches to that. Then you have that first tRNA come in, right? And then we bring in the next tRNA, and we elongate the polypeptide chain. And so finally, it's done. All the pieces go around. And again, this is the end of the story for us. But again, like I mentioned in that previous bullet point, previous slide, then we have this strand of amino acids, and it's not necessarily a protein yet because it hasn't folded into its same shape. So even though this is the end of our story, this is where we end the story. Again, for extra credit, you can look that up. then what happens next, right? Tell me the sequel to this story. And don't forget your sources. If you do turn something in Paris Credit, I need a link to your sources. So that's it in a nutshell. Any questions about that? All right, so again, transcription and translation are how genes control the structures and activities of cells. Because again, for the most part, well, not for the most part, as far as we're concerned, always, this is all about creating proteins, right? Transcription, translation, right? We're going from DNA to RNA to protein. And remember, those proteins very often are enzymes. And because of that, those are the things controlling the activities in this, the structure of the cell, right? It's the enzymes that are in charge of every single chemical reaction, that pretty much every single chemical reaction that's happening in our cell. So that's how that code of the DNA can control everything, right? Things that have nothing to do technically with protein, other than the fact that it's the enzymes, which are made of protein, that direct these chemical reactions. So again, the information originates with a specific sequence of DNA, right? The A's, C's, T's, and the G's, those nucleotides. That's the language, right? That language has 4 letters in its alphabet, and every word in that language is only 3 letters long, right? So you know that there's 64 words in that language. The gene dictates the transcription of a complementary sequence of nucleotides in the mRNA. So again, you need to know that A's and T's go together and C's and G's go together, because you will get at least one test question where I give you a strand of DNA and I ask you to transcribe it, in which case you'll need to pick appropriate RNA choice from the multiple choice question. That mRNA sequence specifies the sequence of amino acids. And again, you'll have a question like that too on the exam, where I give you that big wheel that I showed you before, where you start in the middle and work your way out. So like CGG, oh yeah, that's a leucine. I don't know, I made that up. I have never memorized that wheel. I never needed to, because you always get the wheel to do the translation yourself. And then finally, after you have that polypeptide, which again is just a long strand of amino acids, finally the proteins from those polypeptides determine the appearance and capabilities of cells and organisms. Because again, it's not, you know, it's very much a lot of times enzymes are what these proteins are, right? Not always, but sometimes or very often they're making enzymes. And again, those enzymes are responsible for, think about like the enzymes, even though we didn't name them, think about all the enzymes involved in photosynthesis, right? Without those enzymes, you wouldn't end up with glucose, right? So this, in that sense, that's how your genes control how much glucose you make, or not we, but plants, right? Because there's a gene or genes responsible for making those enzymes, which were then responsible for all those different steps of photosynthesis. Anyway, any quick questions about that side? All right, here's where it changes up a little bit. We used to think that DNA to RNA to protein was the only way things worked. And for the most part, for the exam anyway, you guys consider that to be true, right? We can consider, to keep things simple, it does go from DNA to RNA to protein. We used to think that was the only way that genetic information controls traits. Again, for us, for the most part, you can, for the sake of the exam, to keep things simple. But there have been recent discoveries that point to more complex roles for RNA. So if you read chapter 11, you can learn about that. You definitely don't need to, but it is relevant because there's a lot of new medical technology coming out of this. And unfortunately, again, one of the reasons this course is relevant to your everyday life is one day you or somebody you love might have some sort of bad disease. You need to do some research on how that disease works and how they treat it. And very often you might be coming into some of this stuff, right? Some of this new RNA technology. So look into it if you want. Any questions about that? All right. So for the most part, we're done talking about this whole idea of DNA to RNA to protein. We're done talking about how it works, but now we're going to talk about how it doesn't work. We're going to talk about when things go wrong. We're going to talk about mutations. Now, this bullet point is very important because without understanding mutations, you cannot understand natural selection, right? And natural selection is where we're going after this, or any evolution for that matter. Without mutations, we would not have evolution. Therefore, we wouldn't exist. So this is important. This is not just like, all right, we're going to learn this in this chapter and then leave it behind, right? This is going to be important for later. So let's talk about mutations. Your next word for attendance, Bob Ross, painter. Today is his birthday. Oh, I didn't even take attendance. So I guess it doesn't matter if you guys are sending the attendance words. But I will really quickly for my, since I'm recording for my own records. We've got both Hills here, Brumfield, Sigmund, Perez, Wilson, Gunther, and Smith. So I know. All right, anyway, mutations. A mutation is any change in a nucleotide sequence of the DNA, any change. So far, we have talked about mutations before, and it's going to come back. I'm going to use that same example that I've used before. It's just been one example. And that example I've been using has only been one change. It's been like one swap of a nucleotide. And that is still considered a mutation. But it can also involve large regions of chromosomes. So it doesn't have to be one small mutation. It can be huge differences. And again, it could be a single nucleotide. And again, the example I've been using has been sickle cell. And I told you sickle cell will come back again. It is. I'm going to talk about it again. And then again, when we talk about natural selection, sickle cell is going to come back again. So this is a very common example. But anyway, so again, mutations can be very big. They can be tiny. They can be anywhere in between. Any questions so far? All right, here's an example of a very simple mutation. Right here we have, again, this is the one, this is a real example. This is for sickle cell. Here we have this huge gene. We've zoomed in to a small part of the gene. Specifically, we've zoomed into this one codon. You know, it has a lot of codons, but we're only looking at the one codon that got mutated. This is what a normal hemoglobin DNA looks like. So unless you have sickle cell disease, when you look at your genome and you look at this, codon, you would have a CTT. Anybody who has a sickle cell mutation would have a CAT, right? So we've swapped a T for an A. And because of that, you have a different mRNA codon. And because of that, remember, an mRNA codon says, hey, bring me this specific amino acid. In this case, you should have that amino acid, but because you changed the word, you changed the codon, now you have that amino acid instead. Just that one change now causes you to have one different amino acid. And just that one small change causes you to have that whole disease that we mentioned earlier when we talk about sickle cell and all the symptoms that go with it. But anyway, here's an important part for the exam. You need to know the difference between these two things. There are two major categories of mutations. There's substitutions. There's one category, and then there's deletions / insertions. You're going to need to know those. You need to know the difference between the two. We're going to talk about the details, but for now, there you go. There's the names of them. Substitution is one category, and then deletions / insertions. Substitutions is what we've already talked about. So that example I just gave you for sickle cell, that's a substitution, right? There was supposed to be a T. You substituted it with an A. That is the substitution, right? But let's give you another example of a substitution. It's going to look pretty much the same, right? Here we go again. We're looking at a random gene. We've zoomed in, so we're only looking at 1, 2, 3, 4, 5 codons, right? So here's 5 codons of this gene. This particular codon, instead of being GGC like it should be, we have substituted a G for an A, so now we have AGC. So now instead of having this GLY amino acid, we have this SER amino acid instead. And again, like I mentioned earlier on that last example, it might seem small and insignificant. It's like, okay, what's the big deal? Just one amino acid is different. But that's why sickle cell is such a great example of that, because just having that one different amino acid gives you that whole cascade of symptoms, right? All those complications that come along with that disease, all because you swap one amino acid. But anyway, that's a substitution. Any questions about substitutions? All right, here's where it gets a little bit worse. Deletions. Deletions are very bad. So again, here we have a gene with 5 codons. This is what it's supposed to look like. Here we have a mutation. Here, instead of having UUU, like it's supposed to be, we've accidentally deleted one of those U's. So now, instead of having UUU, we have a UUG. But, and this is where it gets worse, because of you've deleted that thing, everything is shifted down. So now, instead of having a GGC, we have a GCG, right? So that's 2 different amino acids. And now, instead of having a GCA, it goes on and on. Like, so imagine, again, this is only 5 codons. Imagine this going on for hundreds of codons all the way down here, right? Everything downstream of this is going to be messed up. Every single one of those amino acids is probably going to be different now. So if you can have such disastrous effects like sickle cell disease because of one amino acid, Imagine how bad it is when you have just a whole strand of amino acids that are the wrong one. Any questions about deletions? All right, the opposite of that, in a way, as you might imagine, is insertion, right? So delete it, take it away. The opposite is putting one in. In that sense, it's the opposite. But in another sense, it's very much the same. Meaning, this is still just as disastrous as a deletion. Because once you squeeze that new codon in there, excuse me, that new nucleotide, now you've shifted everything that way, right? So now again, try to visualize in your mind, this goes on for hundreds of codons. All of them are going to be messed up because of that one insertion. So again, that's a more disastrous effects than substitutions. So there may be a test question about that. Like, hey, which one's worse generally? Substitution or deletion / insertion? And the answer is a deletion / insertion, right? Because I've explained why. And there's actually a word. Everything I've described has a word. I'm going to teach you that word here shortly. But for now, I just want you to understand the concept of you take one out or put one in, you're really messing everything up downstream. Now I'm going to complicate things a little bit more. There's some other words you need to know. Missense. The missense mutation is a change in amino acid coding. So far, every example I've given you, whether it was a substitution or an insertion or a deletion, that's also, in this case, just the examples I gave happen to be missense mutations, right? Because we had that change, whether it be an insertion or deletion or substitution, and because of that, we changed the amino acids. That was a missense mutation. However, it could do something different. For example, if that mutation were to change it, instead of saying this amino acid, you're getting this amino acid, sometimes, think about it, remember there's 3 codes that say stop the whole process. So there is a possibility that when you have that mutation, whether it's an insertion or a deletion or a substitution, you could change one of those codons to a stop codon. And that is called a nonsense mutation. And you're going to need to know the difference between the two. And it's not hard to know the difference, I think. The hard part on the exam is going to be memorizing which is which, because they kind of sound similar, right? Missense is when you change, you get a different amino acid. Nonsense is when you stop the process. As a matter of fact, there is definitely a test question about that. And it says something like, what happens if there's a nonsense mutation in the code and there's a premature stop codon? What are the results? And the answer to that question, and that is a question, I'm telling you. The answer to that question is it would result in a shorter polypeptide. That's the answer to that question. If there's a mutation that gives you a nonsense, if it's a nonsense mutation, it's going to be a premature stop codon. And if you're going to stop the whole process early, that means you're going to have a shorter polypeptide than you should. So, any questions about those 2 terms up there? All right. So, like I said, when I was talking about insertions, and deletions, I said there's a word for that. I didn't give you the word. I was just explaining the process. Now here's the word. These are called frame shift mutations. Because remember, when you take one out or put one in, you've shifted everything downstream from that, right? So it's a frame shift mutation. In my opinion, that was a little bit easier to memorize for the exam than the other 2, because again, the other 2 kind of sound similar, missense, nonsense. Frame shift, that's completely different. And like I mentioned, they often have disastrous effects. Because there, you're not just changing one amino acid, you're changing a bunch of amino acids. Usually, they produce non-functioning polypeptides. Any questions about that? The next slide I'm going to show you is a picture of a frame shift mutation. This is not from your textbook, I don't think. But anyway, here's the original DNA bases. There's the, we have codons. Those are the codons. These are the amino acids that should be resulting from that DNA code. But here we had a frame shift mutation. So instead of reading this, instead of those being your codons, we moved everything by 1, right? So instead of having CAT, we now have ATT. Instead of having TCA, we now have CAC, right? So we've changed everything. And again, imagine hundreds of amino acids down this way. It's a PowerPoint, so we can't show that, right? But imagine that just goes on and on and on. And now it's completely different. So to summarize everything I've talked about as far as these different categories, this is a great thing to study for your exam. I don't think it's in your textbooks. If you want to study it, download the PowerPoint. But it's great because it's all in one one table, which always helped me as a student. So again, the type of mutations, we have substitution is one category versus insertions or deletions, like we talked about. So substitutions, here's another new, one more word that I haven't mentioned yet. You need to know this one too. There's another effect called a silent mutation. So a silent mutation is when you change one of the nucleotides, right? Because that's what a mutation is. But it doesn't change the amino acid. So if you remember that table, or that big circle that shows you how to translate, remember some amino acids, many amino acids, have more than one codon for it, right? So there's a few words, for example, that might say ribuleucine, a few codons, right? So therefore, if you were to change one amino acid, or excuse me, one nucleotide, you might have a different codon, but that different codon might code for the same amino acid. That would be a silent mutation. And of course, like we already talked about, missense is when you get a different nucleotide than you're supposed to. Nonsense is when you have a stop codon, which stops the whole thing, which results in a shorter polypeptide. And of course, like we said, insertions or deletions are often disastrous because those are frame shift mutations. They change everything downstream from those mutations. Any questions about that, about the different types of mutations? Okay, and then I guess the next question is then, how do these things happen? How do we get mutations? They happen in a number of ways. Sometimes there's errors in the DNA replication or recombination, right? So we've already talked about that. When we talked about S phase of interphase, we did say it's very accurate, but sometimes things go wrong. When we talked about mitosis, we said it's very accurate, but sometimes things go wrong. When we talked about meiosis, we said it's very accurate, but sometimes it goes wrong, right? So if anything goes wrong in those processes, then you have a mutation. Then this thing is called mutagens. These are basically chemical and physical agents that cause it, right? So radiation, some of the chemicals in cigarette smoke, UV light, right? This is why you're supposed to protect yourself from the sun. Like you need sun, but too much of it can damage the UV in the sunlight can, destroy your DNA, cause mutations. So anyway, any questions about how we get these mutations? All right, and to remind you, since you've learned about cancer already, cancer is basically due to mutations. Basically, cancer is very complicated. I'm very much oversimplifying for a 100 level course, but basically that's what it's due to. So remember when we talked about cancer, we said there's all these little proteins that are in control of the cell cycle, like going from G1 to S phase to G2 to the mitotic phase. We had all these checkpoints, right? And they were all controlled by proteins. So if you get any kind of mutation in any of those proteins, and now you know how these mutations happen, that's how we can get problems. That's how we can sometimes get cancer. Again, I'm not saying every mutation causes cancer, but I'm saying generally speaking, cancer is caused from mutations. Or, no, that works too. Sometimes you inherit a particular allele that makes you higher risk for cancer. But that is because somewhere down the line in your ancestors, there was a mutation, and that just happened to be passed along. So, mutations can often be harmful, obviously, but sometimes they can be beneficial. Again, once we start talking about natural selection and other types of evolution in the next chapter, this is going to be very important. Without these mutations, we wouldn't have all these different versions of genes. We wouldn't have all these different versions of life. Everything would just be same basic first life form. So again, sometimes these mutations can be beneficial. If you want to, you can look up some examples for that. Especially with humans, right? There's going to be a lot of humans. Humans are very much studied. Like, what are some examples of when something went wrong, where we had some kind of mutation, whether it be big or small, and it actually benefited us? Anyway, that is it for the second bullet point. We're finally getting into the third bullet point. Before I do, I want to check to make sure we're still online. And while I'm doing that, does anybody have any questions about the second bullet point? Still recording, still online. Glad to see it. All right. Bear with me. Sorry. Just making sure I'm in the right spot here. All right. Anyway, let's get back to it. So now we're finally at the third and final bullet point of this chapter. Right, we're going to talk about viruses and other non-cellular infectious agents. In other words, viruses and other things that can affect you or infect you that are not alive, right? If they're not cellular, then they're not necessarily alive, or they're not alive. So let's talk about it. The next word for attendance is Homer, like Homer Simpson. Today is the birthday of the guy who does his voice. And again, I know I'm repeating myself, but some people are still aren't doing it. Even if you're here, you have to send those words in by 9 a.m. to get your credit. Anyway, viruses. Let's talk about them. When we first mentioned, way back in chapter 1, these properties of life, we talked about the fact that viruses are very interesting because they're almost alive. They have so many of the properties of life, but they don't have all of them. So here we're going to talk about how they are similar. They do have genetic material in the form of nucleic acids, right? So they have DNA or RNA, depending on what kind of virus it is, just like we do. And their DNA is A's, C's, T's, and G's, just like ours. Their RNAs are A's, U's, C's, and G's, just like ours. We also have our nucleic acid packaged in highly organized structure. So do viruses. Of course, type of virus depends on what kind of organized structure it is, but we'll talk about that in the upcoming slides. But again, viruses are not considered alive because, first of all, they're not cellular. And that was the first number one thing we talked about when we're talking about the properties of life, right? Having order / being cellular. And the last one, the big one, they cannot reproduce on their own. You could almost think of your cells as 3D printers. And a virus is like somebody hacking into your 3D printer to get your, get your 3D printer to print whatever code it has, right? It's like sneaking in and like putting your little thumb drive and it's like, I want you to build this instead. And that's basically what a virus is, right? A virus has these codes, these nucleic acids, whether it be RNA or DNA. And our cells, they just do what they're told, right? So they just sneak in that code. The cell's like, oh, look at it. I'm going to build that. Anyway, virus. The definition of a virus, anyway, according to, I think, the old textbook, not that I'm going to ask you the definition, so don't worry about that. It's an infectious particle consisting of genes in a box. It's a nucleic acid wrapped in a protein coat. You'll see what I mean when I show you some pictures of some examples. They're all wrapped in protein coats. Granted, some of them have other things outside of that, but at the very least, they have the nucleic acid wrapped in a protein coat. And again, they cannot reproduce on their own. Therefore, they have to infect a living cell. Which begs the question, if they cannot reproduce on their own, and since they're technically not alive, what you could look up for extra credit is how long can they survive outside of a host? And I'll go ahead and give you a heads up. The answer does depend on the type of virus. Right? Some of them don't live that long. Some of them are really easy to kill. Some of them not so much. But anyway, here's a picture of what we're talking about. There's a box of proteins with genetic information on the inside. In this case, it happens to be DNA instead of RNA, but it could be either one, depending on the virus. And speaking of depending on the viruses, now this is the important part for the exam. There's going to be different types of viruses I'm going to teach you about. Now take a look here at this list. I'm sure somebody can answer this question. This first one that I'm going to teach you about, what do you think it attacks? Bacteria. Thank you. Yes, bacteria. Last semester we sat there for a few minutes before someone answered it. So yes, good, bacteria. Remind me when you send your words in that you answered that question correctly. I'll give you a little bit more extra credit. So yeah, bacteria, viruses that attack bacteria are called bacteriophages. Or sometimes they just call them phage for short. Or some people pronounce it phage. Whatever, it doesn't matter. Point is, these are things that attack bacteria. Now for them, they always consist of a DNA that's enclosed in a structure of proteins, which is interesting to me because the virus, bacteria are very simple compared to us, compared to eukaryotes. But when you look at their viruses, they look a little bit more complicated, right? That looks like some sort of machine. And in a sense, it kind of is. This is basically like a hypodermic needle, right? This is the hypodermic needle, and this is the stuff that it's going to inject. Because here's what it looks like. Here's an actual scanning electron microscope picture of 3 of them. You can see they've attached themselves to the bacteria and they're just injecting their DNA into the bacteria. So that's the easy part. Bacteriophage, they kill bacteria. Right? That's easy to remember. Here's a picture from the old textbook, I think. Maybe your textbook, I don't know. But this is a great visualization. You can see this thing's coming in. You don't need to worry about the name or the fact that this is E. coli, right? It's a bacteria. It just so happens the specific species is E. coli. Anyway, it comes in and it injects its genetic material. In this case, they've colored it red so you can keep track of which is which. The bacteria's genome is colored green. So you can see what's happened here is this DNA comes in and actually incorporates itself into the bacteria chromosome. And remember, for bacteria, they have one round chromosome, right? We have 23 pairs of linear chromosomes. They have one round chromosome. Anyway, you can see here that it incorporates itself into there. And sometimes we get more RNA coming out. And all that stuff gets translated into different proteins. And basically, what they're inserting in there, that genetic information is instructions on how to build a protein. Excuse me, instructions on how to build a virus. So this thing injects its instructions on how to build itself. Then the cell follows those instructions and builds more of itself. And even though we don't talk about this, I don't think in this chapter, and I'm definitely not going to ask you questions about it. Just as a side note, we actually use this a lot in biotechnology. So sometimes when we want to build a particular protein, we take a virus, we humans hijack viruses, put the information we want inside the virus, and then let the virus do its thing and infect these different bacteria cells. And then those bacteria cells follow those directions. They build the stuff we want to build, and then we harvest it. So, it's interesting to me that now, obviously viruses can be bad for humans, but at the same time, humans have learned to hijack them and use them for our advantage. But anyway, all those things I just showed you, it gets a little bit more complicated. This is where it gets a little bit harder. You're going to need to know the difference between these two things. There's a lytic cycle and a lysogenic cycle. Before I even explain how they work, I'll give you my advice on how to memorize the difference between the two of them for the exam. Lytic versus lysogenic, right? Lytic is a shorter word than lysogenic. So I always remembered as a student that the lytic cycle was the short one and the lysogenic cycle was the long one. Once you memorize that, everything else falls into place. So let's talk about it. So again, overall, whether it's lytic or lysogenic, we have this viral DNA replication. Oh, excuse me. This might make more sense when I'll just show you the picture instead. First of all, if you download the PowerPoint, everything I'm about to tell you, you can watch an animation of it. There's one that shows the whole lytic cycle. Here's one that shows you the lysogenic and the lytic cycle. But instead, let me show you a picture. This is from the old textbook. So if the picture in your textbook doesn't make as much sense to you as this, then this is what you should be studying. This is the lytic cycle. It's very short. It's very simple. It's exactly how I described it previously when I showed you that other picture. The virus attaches itself to the bacteria. It inserts its DNA into the bacteria. So you can see in blue, there's the bacteria DNA. In green, there's the virus DNA. They've kept separate in this instance. But again, that DNA, that's just a code on how to build proteins, right? And in this case, the instructions it's providing is how to build more viruses. And that's exactly what it does, right? It goes in there, the cellular machinery, right? The ribosomes and tRNAs, they all come together. They build all the parts to make new viruses. And they build and they build and they build and they build until finally it's so full it just explodes, right? The whole cell explodes, and the word for that is lysis, right? It explodes, and then those viruses are free to go and infect the next bacteria, and the process continues. That is the lytic cycle. It's short. You need to know that for the exam. It's shorter than the other one. Because basically, and you'll see this will make a lot more sense when I show you the next slide. This is the one that's basically relatively immediate death, right? This one's like, all right, we're attacking, we're building a new protein, boom. Excuse me, a new virus. So that's the lytic cycle. Now this is the longer one. This is the lysogenic cycle. It looks similar at first, right? We have the virus attacks the bacteria. It inserts its DNA. But you can see here, sort of, it takes a while to show it. But here, remember, this is what it looked like in the last one. We had the bacteria DNA and the viral DNA.
But in this case, in the lysogenic cycle, and this is one of the questions you might get for the exam, you need to know this one, this is unique to the lysogenic cycle, not the lytic cycle. What happens then is that bacteria DNA incorporates itself, the viral DNA incorporates itself into the bacterial DNA. So now, and that the word for that is called prophage, right? So now instead of having two separate chromosomes, right, they've all merged together. So now, every time that bacteria goes through mitosis, it makes a perfect copy of its one chromosome. It's also making a perfect copy of that part of the chromosome, right? Because that viral DNA has incorporated itself into the one chromosome that the bacteria has. So it's going through mitosis. Every time it reproduces, it's reproducing with that viral DNA. And you can see it hasn't been killed yet, right? So that's the lysogenic cycle. It keeps going and going and going. It's longer, right? It keeps happening. Eventually, however, the lysogenic cycle does kick into the lytic cycle. And then, one of these bacteria that have the prophage, eventually they do become deadly. And instead of just having that dormant DNA in their chromosome, eventually they read the DNA and, the code again says make all these copies of the virus and make a bunch, the cell explodes, the whole process continues. So again, for the exam, you need to know the lysogenic cycle is the longer of the two. You need to know that lytic cycle is the one that actually kills it. You need to know that the lysogenic cycle is when the viral DNA incorporates itself into the bacterial DNA, and the name for that is prophage. So a lot of information on that side. Any questions about it? All right. We'll move on to the next type of virus. This time I'm not going to even ask. So since bacteriophages attack bacteria, as you can imagine, plant viruses attack animals. No, I'm just kidding. They attack plants. One thing you might need to know about them is that they're usually RNA. I'm not quite sure if I'm going to ask you that or not. So remember the one I just showed you, the example I just showed you for the bacterial virus was a DNA virus, which brings me to another extra credit thing if you want to look it up. Don't forget your sources. The example I gave you for the bacteria virus was a DNA virus. So my question to you is, are there any RNA viruses that attack bacteria? You can look that up. Don't forget your sources. Anyway, for plant viruses, most of them are DNA. So for extra credit, you could look up and tell me which ones are RNA. What are the few DNA plant viruses? If you want to look that up for extra credit. Don't look it up now. Do it later. Pay attention to the lecture now. Anyway, some things you should know about plant viruses. They usually stunt plant growth and diminish crop yields, but they're not usually deadly. So they usually don't kill the plant. As far as we're concerned, to be selfish, humans, right? We usually very often require plants for what they produce. And when they get these viruses, very often it diminishes what they produce. One famous example is this one. I'm not going to ask you any questions about it, but the TMV, tobacco mosaic virus. It is a rod-shaped virus that has a spiral arrangement of proteins surrounding nucleic acids. So the picture right up to the top right, that is the TMV. It's hard to tell, but that purple stuff, it's like this. That's the protein, right? That's the spiral arrangement of proteins. Inside, that's the genetic material, the RNA. This particular virus affects tobacco, as you might imagine. It's the name tobacco mosaic virus. But it also attacks related plants, which is another great extra credit question. Which related plants? So which plants are related to tobacco that this infects? And you might be surprised unless you're familiar with these things, but a lot of them are important to us, not just the tobacco. Anyway, it causes discolored spots on leaves, and it was the first virus that was discovered. Not that you need to know any of that, but the textbook included that information, so did I. And again, another extra credit topic here. It says it was the first virus discovered. Who discovered it? When did they discover it? Where? All those typical questions. Any questions about the slide before I show you some pictures of the TMV? All right. So that's what a tobacco leaf that's affected looks like. This is from the old textbook. This is one of those few times where I think the old textbook screwed it up. Because how many of you have seen a tobacco leaf before? Not like in a cigar or a cigarette, but like an actual leaf. Have you guys seen a tobacco leaf before? Yeah, me either. So Well, them showing you like, hey, this is what a diseased leaf looks like. I'm like, okay, means nothing to me. Show me what a regular one looks like so I can have a point of comparison. Like, I don't know. They just think everybody knows what tobacco leaves look like. But anyway, that's an infected leaf. Here's what the whole virus looks like. And then if we zoom in on this part, again, we can see that spiral proteins. But if you see here too, the RNA is also spiraled in there. This is all information that's included in the textbook, so I'm including it. I'm probably not going to ask you to do this on the exam. Obviously, any virus to get into the cells of anything has to get into the cells, right? And for a plant virus, it has to get through the plant epidermis. So like the skin of a plant. Obviously, it's not skin like ours, but it is like ours in that it protects it, right? So it has to get through the epidermis. This is why damaged plants are more likely to be infected, right? So I'm sure you've seen you're walking along, you accidentally hit a leaf, you break it, that's a damaged plant. It's now open, it's now exposed to viruses. Or farming tools, right? So if people pruning things, like you're cutting things down, you're damaging the plant, you're exposing it, it's a wound, it's exposed to the elements, it's exposed to viruses. And if you're using a farming tool, if you cut one plant that's infected and then go cut another one, then you spread the virus. Another thing to note, and I'm not going to ask you this either, there are no cure for most viral plant diseases. And you can look that up. Why is that the case? Correction credit. Why is there no cure for most plant diseases? Conversely, which viral plant diseases do have cures? And what are the cures? So many questions. Anyway, since there's no cure for most of them, agriculture scientists mostly focus on prevention, right? How do we prevent the disease to begin with? And how can we breed versions of these species that don't get infected as easily, right? So sometimes it's breeding, traditional breeding. Sometimes it's genetic GMOs, right? So like, oh man, we figured out that this virus attacks this plant by this means. So let's swap out this gene for that gene. So now it's not susceptible to that virus. So any questions about this relatively unimportant slide? And I say unimportant because I mean, as far as your exam is concerned, that's unimportant. Obviously, for our everyday lives, this is very important because agriculture feeds us. So anyway, no questions? Oh, excuse me. Thank you. Speaking of viruses, I might have a coronavirus. Not the coronavirus, just one that the cold. The common cold is a coronavirus. Anyway, let's talk about animal viruses now. Now we're getting there, right? Now we're getting a little bit more in our wheelhouse since we are animals. Obviously, these are common causes of diseases. And still to this day, I think including COVID, even though this was written before COVID, I still think this bullet point is true. No virus in the human history has had more of a threat than flu. Flu virus, which seems odd to us now because, probably everybody in here has either had the flu or know somebody who has. I know I've had the flu. It sucks, but it didn't kill me. But still, it's been very deadly in the history of humans. Like many animal viruses, the flu virus has an outer envelope of a phospholipid membrane that has spike proteins. So remember, when we talk about those plant viruses, it was just protein and RNA. When we talk about those DNA viruses, protein, and then inside of it was DNA. Here, it's a little bit more complicated. Here, it has basically a cellular membrane. We don't call it a cellular membrane because technically it's not a cell. Therefore, technically it's not a cellular membrane. But for all intents and purposes, it is. It's just like our cell membrane. It's a phospholipid bilayer that has things sticking out of it, just like ours does. Now, the things sticking out of it are a little bit different, right? So you can see these little protein spikes. And again, like I mentioned, I think Monday or last week, even though we're talking about a different virus, this is basically how the COVID-19 virus, mRNA virus works. It codes for one of those proteins, right? So when you take that, vaccine, that RNA, that mRNA says, hey, make that protein. That protein in itself is completely harmless. But once you inject that into your cells and your cells start making it, your cells start recognizing it as foreign, and your immune system is then trained to fight it. That way, when you get, again, this is flu, but let's pretend like we're talking about COVID-19. When you get that virus inside of you, even though it's never seen the virus before, your immune system has seen that protein before. It's like, oh, I know how to fight this. And it attacks it. And hopefully that either keeps you from getting COVID-19 or at least lowers the effect it has. But anyway, that's just that's just the flu virus. This is just a great example, but I'll put a big old X through this because I'm not going to ask you any questions like what's the most deadly virus in the history of humans, or what does the flu have a membrane or not? Right? I'm not going to ask you any questions about this. Just an example. Many animal viruses have RNA. So again, when we talked about bacteria viruses, those examples were all DNA viruses. When we talked about plants, all those examples were RNA viruses. And now here I'm telling you for animals, we can have either. There's RNA and there's DNA. I'm not going to ask you to know which is which, but some RNA examples include the flu, like we already talked about, the common cold, which again, that's a coronavirus, which is in the same category as COVID-19. So this was made before COVID-19, but that would be in that category, right? Measles, mumps, AIDS, polio, all of these things are RNA vaccines. They're RNA viruses. And we have vaccines for a lot of them. Then we have DNA viruses, and we're going to talk about some of these separately. But DNA viruses include hepatitis, chickenpox, herpes, things like that. Yeah, those are just examples. Again, this is not a health class, so I'm not going to, there'll be no questions where I say, which one of these is not a DNA virus? Or which one of these is not an RNA virus? You don't need to memorize that. Would be a good extra credit question, though, but I don't plan on giving you that as a regular question. Here's a drawing of the, like I said, the last picture I showed you was a little bit more realistic. This is more of a less realistic, but it's easier to visualize different parts. So again, with these viruses, these animal viruses, they have that membrane on the outside that helps them merge into the plant membrane. Because think about it, I mean, the animal cell membrane, because animal cells don't have plant walls, right? So we can just, this is why it's probably advantageous for them to have a membrane on the outside so they can merge with ours. But this should look familiar too. Again, this example happens to be RNA, sometimes it's DNA. But look, hopefully you remember what I showed you previously. That looks to me like a bunch of plant viruses wrapped up in a membrane. And it is, in some ways it's true, right? They're very similar. They have their similarities. Anyway, when we get back on Monday, we will I'll show you a generic schematic diagram of how these things affect your cells and how it works. So don't forget, send those words in by 9 a.m. And not last word, but don't forget you have to include one question about the material percentage in the sector. And that's it. You guys have a good Wednesday. I'll be here for lab if you need me. You don't need to be. I'll also be online for lab if you need me. And don't forget, you can wear Halloween stuff to class on Friday if you want. I haven't decided if I'm going to or not. Please just. Oh, sorry about that.